Viruses. Viruses are sometimes classified as microorganisms because they are microscopic and carry nucleic acids. Yet most scientists do not consider viruses to be microorganisms because they are not alive. They cannot reproduce independently. They must use the machinery of living cells to propagate. Viruses move between cells in the form of virus particles. Viruses may infect bacteria, plant, animal, fungal, or protozoan cells. Note that viruses may infect other microbes, and the microbes in our bodies are carrying many viruses. All virus particles have a nucleic acid genome, either single-stranded or double-stranded, RNA or DNA, a protein capsid, made up of proteins called capsomeres, surrounding the genome. Some virus particles may also have a lipid envelope derived from the host cell plasma membrane, as in RNA viruses, and a sheath-in tail, as in bacteriophages. Most virus particles are much smaller than the smallest bacterial cells. Particles range in size from about 20 nanometers, or 2 times 10 to the minus 8 meters, to 500 nanometers, or 5 times 10 to the minus 7 meters. Because viruses are so small and intracellular, we can't see them in our environment. Click on the link to see for yourself the relative sizes of viruses. Viruses are classified based on their genome structure, their capsid structure, and whether or not they have a lipid envelope. They are not found on the phylogenetic tree of life because they are non-living. Like organisms, viruses do mutate and evolve, and their evolutionary history is documented in their genomes. However, viruses' genomes must enter a host cell in order to be replicated. Entire virus particles may enter cells, or they may inject their genomes into cells. Once inside cells, the viral genes can control cell function and direct the production of more virus particles. Viral genomes may be single-stranded or double-stranded, and may be DNA or RNA. Examples: coronaviruses such as COVID-19 have single-stranded RNA genomes. HIV particles have 2 copies of a single-stranded RNA genome. HPV, human papillomavirus, the virus that causes cervical cancer, has a circular, double-stranded DNA genome. The viral capsid surrounds the viral genome. The capsid is made up of proteins called capsomeres arranged in a repeating pattern. Most viruses have capsids that are either icosahedral, 20-sided, or spherical. Virus capsids have a range of shapes. Some common shapes are shown on the following slide. 4 basic shapes of viruses are shown here, including the one labeled as A, which is a helical capsid. An example would be a tobacco mosaic virus. The one labeled B, that's icosahedral capsid. An example would include adenovirus. The one labeled C, multiple helical capsids, spherical viral envelope. An example being the influenza virus, the virus that causes the flu. And the one labeled D, the complex capsid, an icosahedral head and helical tail, and the example is a bacteriophage. What are the spikes of a virus particle? What are the spikes of a virus particle? Some virus particles are enclosed in lipid envelopes that are studded with viral-encoded glycoproteins called spikes, as shown in the image on the previous slide. These spikes are the structures that bind to cell surface proteins. They are the virus particle's landing gear. When they bind to a cell surface receptor, the virus begins its penetration of the cell. Note that these spike proteins have 3-dimensional shapes. These shapes determine the cell structures to which the virus can bind. Virus infections are usually specific. They bind to only one or a few receptors found on a limited number of cell types in a limited number of species. The shape of the spike protein is an important factor in determining the host range of a virus. Spike proteins are often antigenic in animal cells, meaning that they induce immune responses. Spike proteins provide antigens for the immune system, and our immune system's killer cells and antibodies bind to them. For example, the SARS-CoV-2 COVID-19 spike protein binds only to a protein called ACE2, which is found on human epithelial cells in nasal passages, lungs, and intestines. This distribution limits the virus's route of infection. The Moderna and Pfizer COVID-19 vaccines are RNA molecules that encode modified spike protein genes inside a lipid vesicle. For reference and further information, visit the website shown here. Bacteriophages. Bacteriophages are virus particles that infect bacteria. They have complex structures with a head and a tail. The head is a capsid containing the viral genome. The tail is made of proteins that bind to bacterial cells and inject the genome into the cytoplasm. The animation GIF at the link below shows the process of bacterial infection by a bacteriophage. Notice that the virus injects its DNA into the target cell, but does not enter the cell. Click on the link here to see a little GIF of bacterial infection by a bacteriophage. How do viruses replicate? Viral replication varies tremendously, but in all cases, a virus infects a cell much larger than itself and directs the cell's metabolism to produce more virus particles. The cell usually dies as new viral particles emerge from the cell. Here's a general pattern of viral replication. 1. The virus particle binds to the cell surface. 2. The genome penetrates. 3. The genome is transcribed and translated using host and/or viral enzymes. 4. New virus particles are assembled. 5. The particles are released from the cell. The animation GIF found at the link below shows HIV binding, penetrating, and being reverse transcribed, as in steps 1 through 3 above. Coevolution of viruses and hosts. Viruses and their hosts apply natural selection pressure onto each other. Viruses are always evolving to infect cells efficiently without destroying their host species, while host species evolve defenses to eliminate viral infections. Our immune systems have evolved to remove or control pathogenic viral infections. Control of viral infections. Some viral infections quietly integrate into host cell genomes and set up long-term asymptomatic infections. This viral adaptation ensures survival of the viral genome. In fact, 8% of the non-coding DNA in the human genome is remnants of past viral infections, passed on from parent to offspring with every generation. The quiet integration of viruses has added to the plasticity of host genomes, allowing them to evolve faster, and helping the survival of host species as well. Some viral infections are pathogenic in humans, meaning that they can cause disease. Pathogenic viral infections can be controlled with antiviral vaccines and drugs. Vaccines induce an immune response to viral antigens before the virus infects a person. The presence of memory B cells, T cells, and antibodies gives an immunized person a head start in the battle against the virus if it infects later. Antiviral drugs typically inhibit enzymes that a virus needs to penetrate, get transcribed, integrate into the host's DNA, or assemble new virus particles. Some anti-COVID-19 drugs and antibodies that prevent attachment of virus particles to host cells.